processes across cellular populations, almost as long as the workshops are.
Right. I should have listed the acronym of my talk, but I didn't. Right. So I thought it was quite charming how everyone was apologizing whenever they mentioned spatial things. I, on the other hand, actually literally put spatial into the title of my talk, so I feel I'm feeling rebellious. To make matters even worse, I'm going to start with a quote from a biologist. I'm calling it a slightly wrong quote from a famous scientist. The dream of a bacterium is to become two bacteria. It's not so much that it's slightly wrong as that it's oversimplified. We know that there's a reality of heterogeneity that means that it's not a simple matter of dividing as quickly as possible. And so what I'm going to try to do is attempt to use this quote from Francois Jacob as a bit of a straw man. But rather than actually knocking it down, I'm going to then attempt to actually find a way in which the way he said this was perhaps insightful for how we should be thinking about analyzing growth processes in cells. So here's the sort of a representation of the growth process in reality. We see that this is contrived conditions that I've set up where the cells are growing under a microfluidic device. They're growing heterogeneously. All we're seeing is the fluorescence channel. It's kind of a terrible device that I'm using because it allows cells to rattle around. So we see heterogeneity. We see, if you watch carefully, there's definitely some inheritance of the growth rate and other phenotypes. This fluorescence is constitutive, so it actually reflects the global physiological state of the cells rather than any kind of gene regulation. There's a constant flow of media, so there's a relatively uniform conditions in which they're growing. And we see that some cells are dying, some are growing quite quickly, some seem to be, some are looking rather chubby and they're growing relatively slowly, and they seem to be inheriting that slow growth from their mother cells. And this black circle here is actually, if I had shown the bright field, you'd see that that's simply a support structure that is made out of silicone. So this is a microfluidic device with a constant flow. And although it may look like it, there is no spontaneous generation going on. This is cells that are wandering in from the outside. So it's maybe not the best movie, but I feel like it does give us an idea of the kinds of dynamics that cellular growth can deal with. And I'm actually going to show how this sort of thing can arise out of threshold-based regulatory mechanisms, which seem to be ubiquitous in controlling bacterial growth rates. So it seems, I'm going to argue that bacterial growth rates have evolved to be intrinsically heterogeneous for various reasons. So how do bacteria grow? It's all about doubling the number of ribosomes that one has. You could, in a sense, think about the entire cell as a support structure in order to get molecules into the cell to produce enough ribosomes to be producing more ribosomes, right? So a lot of the ribosomes have to be committed to producing things necessary to allow more ribosomes to be produced, right? So we have the biomass, we have a constant protein fraction of structural proteins and the things that actually carry out the mechanism of biomass growth and division. So you know, we could think about then bacteria have evolved to grow very quickly, and which sort of makes sense. You know, it's famously they grow exponentially, and there's this you know, famous anecdote that if there were an infinite amount of nutrients and no limitation to their growth at all, that a single E. coli cell would probably be the size of the Earth within a day, or something like the colony resulting would be the size of the Earth within a day. You know, that sort of thing. So that's not what happens, of course. Cells spend most of their time not growing at all. usually because they're resource limited, but not always. It turns out that the slow growth phenotype is useful. So this is a now aging experiment that was done a little over 10 years ago now, where this is a primitive microfluidic device with these channels, and these are single E. coli cells that you're seeing growing. They're rod-shaped bacteria, right? So they grow linearly in the channels, and we can see that there's a cell up here that is not growing at all, and that lack of growth, that growth arrest phenotype, has permitted it to survive antibiotic treatment, whereas essentially the rest of the cells have died and gone away. And then once the growth medium is returned, then the cell starts growing again. So there's, in some sense, a sort of a statistical searching for different phenotypes that may not be optimal in one environment, but could allow the survival of a different stressful environment. And there are practical reasons for why this is important. And so this is a cartoon that I drew more for biologists that sort of shows when bacteria can infect a mammalian host. This is supposed to be a representation of lungs, which I'm quite proud of that it vaguely looks like lungs because I traced it myself. And it's so we have some mutilational event where there's a bacterium starting to attempt to take over your body. Of course, most bacterial infections are cleared out by the immune system right away. If you have certain predisposing conditions, so for example in the lung, if you are a cystic fibrosis patient and you are infected with an organism called Pseudomonas aeruginosa, then they will frequently start to grow out, and then the immunity will kick in, and there will be a process of growth arrest and the formation of biofilm. And what happens here is some of the cells end up actually sacrificing themselves, or just in effect dying somehow, and their cytoplasmic contents are contributed to this gelatinous goo, I guess you could. I don't know. That's redundant, but anyway, so these slow-growing cells end up basically establishing themselves in different parts of the lung, and they're heterogeneous and they're very difficult to get rid of. They're not growing very quickly, but there's I guess in a sense, a delayed reward of having have at least surviving in the host, right? So I'm really interested in the process of when cells are growing very quickly and what effects that the process of growth arrest has on the physiology of the cells when they get there, right? And so when we think about this growth arrest process, so if there's something endogenous within the cell that's causing the growth arrest, it forms sort of an implicit positive feedback loop. Okay, so here's just a very simple cartoon. So it's a toy ODE model of we have two molecular species. They're being degraded in bacteria. Very frequently, proteins are much more stable than the cell cycle time, right? So this degradation event is often dominated by the growth rate. So as the growth rate is slowed down, the dilution is slowed down, and so it ends up being sort of this positive feedback loop. And this has been explored by various people in the past. So Plump et al. Were exploring the fact that if you have a toxin being expressed inside a bacterial cell, so there are many different toxin, antitoxin systems, and endogenous toxins are present in bacteria and do various things that we'll discuss in just a second. If the toxin is somehow inversely proportional to the growth rate. When you have binomial partitioning of the toxin, you get some cells that are effective at continuing to dilute the toxin to keep it at such a low level that cells can keep growing, while others will end up being trapped into this constant buildup growth rest process. So that you can build models that make it look like it's bistable. There are other not explicitly toxic, or not systems that have evolved to be toxic, but synthetic systems. So Lingchong Yu had this this project looking at a synthetic feedback loop that ended up causing bimodality in the cell count in terms of the fluorescence. The level of the fluorescence was directly correlated with the growth rate, and so these cells were slower growing, right? So we've seen these things happen before, and what's interesting about these thresholds is that they seem to be present very often in cases where there's modulation of growth. Okay, so as I mentioned, there are these toxin-antitoxin systems. That slide I showed a few minutes ago with that cell that survived the antibiotic treatment was growth arrested because there was an excess of toxin over the antitoxin, right? So there's this molecular titration effect that can occur where when you have equimolar quantities of the toxin and then its neutralizing antitoxin, the complex is harmless to the growth of the cell. But as soon as you exceed this threshold, there's then a linear increase as you increase the production rate of the toxin. It's not been quite as well appreciated, but this can happen in metabolic pathways as well, and I'm going to talk about that project that I did relating to that. So I'm going to get to that detail later. That it has to do with the enzyme saturation. But then there are cases where there are stress responses, conditional stress responses that depend on global gene regulation. This is the so-called sigma-anti-sigma system. So a sigma factor in bacteria is a subunit of the RNA polymerase. Different gene promoters have specificity for certain sigma factors, and so if an alternative sigma factor, so there's a standard sigma factor that is generally ubiquitous and healthy in standard growth, and then during stress responses there can be alternative sigma factors that are always present in the cytoplasm but are being sequestered by the anti-sigma factor, and when they're released, it then allows a global stress response. Sigma factor, so this is an outdated picture of how we could think about gene regulation in E. coli. So sigma factors are global regulators, so they would be up here on this top tier, and they tend to, and these green ones are the sort of the output, so in a sense the phenotype, the phenotypic response, and these are intermediate regulators, and so sigma factors really cause global shifts in gene expression. The same thing happens with these toxin-antitoxin systems. So there are many different mechanisms, and there are many different toxin-antitoxin systems, even within the same strains of bacteria. So they are oftentimes, the most well-characterized ones interfere with the process of translation. So the genes being transcribed and then translated in that translation process slows down. The cell eventually stops growing. Interestingly, so they can also inhibit phospholipid synthesis, but interestingly recently, and I don't really like this, I don't think this picture is very accurate, but it sort of captures a connection that has recently been made between one of the toxin-antitoxin systems in E. coli, and it seems to be unique to this particular one, and central metabolism. So there's a starvation response called the stringent response that is that results in ribosomes being essentially chewed up so that their amino acids can be used for stress responses and other purposes, and reducing the fraction of the ribosome content slows the growth, and this toxin also has the effect of inhibiting translation. So, and then it, somehow via some unspecified mechanism, the authors are claiming that it also regulates other toxin-antitoxin systems. So the point being that there's a threshold-based mechanism here where when the toxin exceeds the antitoxin concentration, then we have this process of global metabolism basically going through a shift to being shut down in single cells, right? So looking at this molecular titration effect in a little more quantitatively, it looks like essentially that there's no free amount of whatever your effector is, toxin or sigma factor, up until a threshold, and then above this threshold, then as you increase the ratio of the two molecules, it's basically free to take any concentration whatsoever. There's no particular regulatory mechanism stopping it at a given concentration. So it seems to suggest that there's something like a point where there's a kind of a big a generation of a lot of heterogeneity on this side, and then cells that have not committed to any response on the other side of the threshold. So this is a kind of a mysterious type of mechanism, because in the presence of gene expression noise, if one is close to the threshold, you expect that there's going to be a lot of cells with many different possible concentrations of the output. And so what implications does this have for the physiology of the cells? I want to show how this same effect can happen with a metabolic pathway. So we have we'll be exploiting the fact that there's a difference in time scale between metabolite fluctuations and protein fluctuations. So this is a stochastic simulation where we had these two enzymes, A and B. You can see when enzyme A gets quite high, and I'm not showing B here, but when enzyme A gets higher, enzyme B will have a tendency to saturate, and this can cause a very quick spike in the level of the metabolite. So I mean, that in itself is sort of it's sort of a fun little phenomenon. But what makes it really interesting is when the metabolite has some small level of toxicity. Then something really interesting happens. So I tried to set up a model to look into that case. How does so it would seem intuitively that sort of the optimal throughput through the metabolic pathway would be one where the enzymes are nearly saturated. That may not be how they actually operate, but it seems that would be the best way to get a high flux through. And so I wanted to explore how close to that sort of point where you could have a dangerous buildup of metabolite can you get and still have relatively good growth rate, right? So I built this model, very phenomenological, and I'm sort of on shaky ground here because I've assumed that the time scale of the metabolite is much faster than the enzymes. So we have a constant flux, and we're solving for the steady state. But then we have these interactions with the growth rate where the consumption flux is proportional to the growth rate up until we reach a threshold where it's no longer the rate limiting step in the pathway, and then we have a toxicity effect from the metabolite itself. So that's inversely proportional to the growth rate. So these functions ftox and FGR basically determine the dilution rate of the metabolite in addition to the consumption rate of it. So we can use this to infer the growth rate. And I mean, the problem here, right, is that we're sort of saying, okay, a miracle occurs. We're not specifying what the mechanism of affecting the growth rate is. So I don't necessarily have any reason to believe that this is a very good model to begin with, except that something fell out of the model that made these assumptions, I think, all right. And that is that we reached a point where there was in the steady state, there is a singularity that would occur as we increase the ratio of the production to consumption flux. So what happens is that more and more of the metabolite is being produced, right, until we get to the point where the second enzyme saturates, and we reach that a threshold where no more essentially the consumption in this pathway has become something like zero order, and then we have just a constant increase in the amount of metabolite, and so there's a point where it can't return. So I got my slides out of order here, right? So we have a rate of change of the metabolite in the model, and as we change this flux ratio, right? Here's the maximal growth rate for the particular parameters that I'm using for this plot. There's a point where there's a threshold where there's no longer a steady state that exists on this scale, and so if the maximum growth rate is near that, we could imagine that a little bit of noise in the input flux would be enough to send some cells over this edge, this irreversible threshold. And so this is a plot here of the metabolite production rate, right? So we're changing the ratio. We're keeping the consuming enzyme constant, and we're increasing the amount of the enzyme that's producing the metabolite. And there's a regime that's in red where the cells predicted to be growing quite healthily, and in a high demand environment, so an environment where there's a need for a lot of this pathway activity, the size of this region gets smaller and smaller, and this threshold here is one that corresponds to that dashed line. So it gets to the point where there's an irreversible buildup absent any other mechanism kicking in to further clear out the.
Sorry.
Yeah.
You multiplying the F toxin by the F is your growth the product of the effect of the two processes.
Oh, I'm sorry. Yes, that's absolutely what I'm doing. So I have a yes, growth consumption or a growth rate that is the maximum growth rate times F tox, which is bounded from 0 to 1, times the FGR, which is also bounded from 0 to 1.
The result of it is a deterministic model.
This is completely a deterministic model. Yes. So there's no noise here at all. We're just using this as a way of thinking about where the noise might arise somewhat intuitively, right? So we would expect that there's going to be a case here in the very high demand regime where if there's fluctuations up and down a little bit, some of the cells might be starving, some of the cells will be happy, and then there will be some cells crossing over into this what is on the time scales of interest here, an irreversible threshold. Right. So I wanted to see if this, if there's any empirical reason to believe that this could be occurring. So I did it in this E. coli system with just changing the amount of extracellular lactose. The reason why I chose this was because the particular molecular mechanisms of the way lactose is imported into a cell. It brings in a proton with it as well, and so there's a well-known toxicity associated with that. So lactose is a suboptimal sugar for cells to be growing on. And it turns out that indeed we get, so we want to think about the population growth, in the exponential phase. So I basically ran these experiments where I took a plate reader with this polystyrene 96 well plate. I have the different concentrations of the lactose, and it's kind of noisy in the low region, but we can see that there's a distinct plateau in the growth rates, and then it starts going down. So there is a toxicity that is occurring. The problem is that we don't know if this is all of the cells becoming toxified at the same time, or if there's this heterogeneity effect. And oh yes, one important detail I failed to mention was that I grew this only in lactose, so it was a minimal media, and so I sort of forced the demand parameter to be very high in the experiment in order to create the dynamic. And so I'm using this, exploiting this result from Klump et al. Right. So fast-growing E. coli have a larger volume, as Vahid pointed out earlier this week, and the effective concentration of proteins tend to be lower. So what I did was I used this constitutive fluorescent reporter that I showed you earlier in these E. coli colonies to infer with flow cytometry basically what is the fluorescence of the cells. I expected it to be the inverse of the growth rate, or inversely proportional to it anyway. So there's so I did, for all these different lactose concentrations, I went and I broke the record for how many flow cytometry samples were done in a single day on the flow cytometer at the University of Kansas, and I got this nice curve. So we see that at lower growth rates, the cells are on average brighter, the brightness decreases, and then plateaus. And we see that this plateau should be corresponding to where the cells are growing quite quickly. So the average cell in these cultures is growing fast, but the population itself is growing slowly. So how do we explain this? Well, I mean, I think that it should be fairly clear from looking at that phase diagram and using our intuition that there's essentially a metastable growth dynamic going on. So the cells are, there's a exponential growth rate that's fast, but as we increase the lactose concentration, the switching rate into a non-growing state increases. And if we use this very simple two-population model, the growth rate is still exponential in the long run once the population is stabilized, but it's instead of being just G, it's G minus S. And we see that indeed there is an increase in heterogeneity. So, right, so this is the coefficient of variation in yellow, so the cells seem to have more growth noise occurring. I finally actually did some stochastic simulations just using a standard Doob-Gillespie algorithm, and so I created a large model where I included gene expression noise. I included all of the molecular interactions were mass action. So I broke everything down into elementary reactions. The only thing I did that's philosophically questionable here was I did have complex propensities that we used to compute the dilution rate that were the same as the ones that I use in the deterministic model, right? So we're looking at three cases here. This is the peak growth rate. This is the low starvation regime, and this is the when we're above the peak growth rate. And we can indeed see that there seems to be a threshold where most of the cells are living happily below. I should mention that these are independent simulations, all starting from initial conditions of the predicted mean field. So the mean of the system, and then as the cells go across the threshold, notice this is the log scale, right? So we get this long smear of cells at various concentrations. So it seems to be consistent with the view that the cells are growing metastably around this, and there's a lot of heterogeneity, and there's no apparent reason for it other than the fact that there's simply a threshold there. Although I did do an experiment that I'm not showing here that showed that if I take the populations that correspond to this in E. coli and I treat them with antibiotic, the number of persister cells present is greatly enriched. So the cells are probably better at surviving antibiotic treatment, not unlike the toxin-antitoxin effect, right? So we get this heterogeneity in growth rates, and that's fine. But what we find is that at very high concentrations, we're actually seeing a lineage relationship that I wasn't expecting at the time. It turns out not to be unprecedented, of course. So this is a paper from a couple of years ago by Ni et al., where they showed that if you track the lineages and they're tracking a phenotype, a RPOH-driven YFP, so this is essentially a stress response after they've added sub-inhibitory concentration of antibiotic, you see that there are these related sister cells that tend to have higher, correlated levels of stress response. And what's interesting, though, is that in our case, the number of generations required for this correlation to have occurred in this particular case is much larger than what they saw in the previous one. So it made me wonder if we need to explore in a little bit more detail what's going on with this lineage relationship. And so I'm going to pull a little switch here and actually change over to using a toxin-antitoxin system to do this study. And the reason for this is that the metabolic system has a non-monotonic response to our the conditional parameter, which is the sort of what the flux through the pathway is, right? So we increase. There's a starvation regime and a toxicity regime. I got rid of the starvation regime here with the toxin-antitoxin system. So it's a simpler system, and we're going to create an agent-based model of this, and then and basically implement this very simple growth, this interaction here between the scales, where we have agents that are growing. We see that the growth feedback effect in the mean field model also shows the singularity. So we have a point where there is no steady state there as well. This is perhaps shown more clearly here. So we have a toxin production rate we have a toxin that's very low. As the toxin production rate is increased, it reaches the threshold where it equals the antitoxin production rate, and very quickly it goes to, on this scale, it goes to infinity, which of course on the cellular scale corresponds to a much different situation. We probably have some other effects on a larger scale taking over at that point. And so I wanted to try to understand what that was. So with my postdoc, Arna Bandyopadhyay, we created a branching process simulation. It was extremely simplistic. So we basically have production propensities of the toxin and the antitoxin in each cell, binding and unbinding. And then we have a change of the cell volume over time that is a function of the toxin concentration T. And actually, we have a delay here. And we figured that the amount of time for the toxin to start take effect would be however long it takes to percolate up through the network. So at division time, we have binomial distribution of all of the molecules into daughter cells, and the dilution this function is actually dependent not on the toxin number but on the concentration when the cell is born. And we can change this parameter so that it's an arbitrary delay of whatever we wish. But I just want to critique this model a little because it kind of sucks. So we have exaggerated cell cycle synchronization. We have uniform propensities from cell to cell. I kind of like these facts because it means that when we're seeing effects from the toxin-antitoxin system, we really know that that's what it is. right? We've gotten rid of a lot of other reality so that we can just look at the effect of the toxin-antitoxin. The thing that really bothers me about this is that we have an arbitrary relationship between toxin and growth rate. But proceeding anyway, right? So we have this phenomenological scale transition occurring at this interface between the growth rate and the kinetics of the toxin-antitoxin system, which we've implemented, I think I failed to mention, is just a standard direct SSA. So we basically run these simulations where we allow a few generations of outgrowth, then we apply a stress, right? So it becomes, we're putting constant energy into the system, basically producing more toxin level. And the way that the trajectories tend to look is that we end up having these cells, so this is the antitoxin minus the toxin. So higher up, there's more antitoxin, and when we're negative, there's more toxin, and this is over time, and we see that we have these nice synchronized cell cycles. There's a slight toxin production rate, but not very high, and then we, at this time point 100, we start really producing the toxin, and then so the cell keep going down. There's still some growth, some division going on, but it slows down, and it's not. I'm showing a case where it's particularly toxic, but if I show one that's higher, there's a much bigger growth, much more growth. So as we look at this is our whatever the units are here. We have basically you can look at the doubling rate as we increase the toxin production rate, keeping the antitoxin production rate constant, and we end up with this sigmoidal response for the entire population after this certain arbitrary amount of time. It generally, so here's how it kind of looks. Let's see. So this is scanning the toxin production rate parameter over time, right? So, and then up here is the amount of toxin in the cells, and the cells are basically lined up. So there's a vague relationship to how closely related the cells are in terms of lineage. But we basically end up with a case where we have almost no growth arrest, and then going down to where the cells have essentially stopped growing. And as we look through those, and we look at the lineages that we get, we find that there is indeed, as we were hoping to reproduce from the experimental data that motivated this, we see that as the cells, as the conditions approach those such that the cell growth is going slower, we see these clusters of red in the lineage. This is cells with a large concentration of toxin. You can see the effect of the growth rate because the lineage is getting narrower and narrower, so there's less growth. And you can see side effects of the artificiality of the system, right? So here, at very low toxin levels, we have highly synchronized cell cycles. I zoomed in on a couple of them because I wasn't sure how well those would show, but I think we can see them well enough, right? So here we see that there are occasional cells that have started to accumulate some toxin at the rather lower production rate. At the high production rate, we see distinct clusters of cells that have emerged from sort of sub-clades, if you want to call them clades, I guess, sub-lineages of the population. So what I wanted to do was to, okay, I turned the screen off. Okay. What I wanted to do is try to find a way to measure this clustering effect. And the idea was that it felt like vaguely like there's some critical point in the population growth rate, and there's some transition, right, a growth arrest transition occurring. And these things, I thought that we could maybe try to find some kind of a correlation. And so we looked at what seemed to be the most natural way to determine for each pair of cells at the final time point, how far apart are they in the lineage, which could be characterized by how long it's been since their most recent common ancestor. And then we could compare this to the any sort of phenotypic readout we wanted, the difference in doubling time between them, or the difference in the amount of toxin level. It's not clear to me what the right one to really look at is. And what we end up getting is, which I'm not completely happy with yet. So we vary the toxin production, and we see that as the growth rate of the population is in its transition zone, that there is there's a definite peak in the correlation there. And I don't know that this. I tried a couple of naive ways to do this. So we use a non-parametric correlation measure. We looked at the slope, basically fitting all these dots to a line, which just seems really bad, except that it got rid of a lot of the noise down here at the lower toxin production rate. Right. So we know that there's not much going on down here from visual inspection, but there's a lot of noise in the correlations there. The important thing, I think, is as we approach this this high slope area, then there's a peak in the correlation of the lineage space. No inheritance control shows that it depends on the inheritance. But I'm basically leaving this at an unsatisfying point where what is the most appropriate measurement of the growth arrest transition? I don't really like anything that I've done. I want to think about ways to, I want to change to think about how do cells actually measure what's important. And I think that actually has to do with selection, the process of evolutionary selection. So what is the probability in the future environment of the growth arrested cells being beneficial, perhaps? Okay, yeah. Right, so we have, so I was looking at naive things like, let's look at the entropy. My postdoc decided to do it in base E for some reason, but this is how much toxin. Because it's a smear, there's a lot of variability in the high, in the growth arresting region. This green is where we have no inheritance at all. So, I mean, we definitely see that there's a peak in the entropy. So what am I saying here with this? Well, I think that there's some way to measure the heterogeneity of the outcome population and how we can relate that to what the future environment might end up being. And I have like 30 slides I'm just going to skip through now and go on to something else. Right, so anyway, the point being that this quote from Francois Jacob is actually, he's sort of viewing it from the perspective of the bacterium as opposed to from the perspective of the experimenter, and I think that that's a good way to do it, and I want to find a way to try to understand this growth arrest transition in the same way. So I don't have any, I was going to talk about one other thing as well that I think I'll give only very briefly. So we had a lively discussion over lunch, some of us did, about this. Bennett in 1982 was thinking about how to optimally compute on so this is like a Turing state space, and moving down the line of a deterministic outcome. A bacterium would, in some sense, be like in the reverse of this, right? So it would be occasionally sampling spaces for an unpredictable future statistically. So it might be sort of like a statistical computation thing. And so we could imagine that there would be all these different state spaces that could arise. And the question is, for a bacterium, you want the size of the state space you want the phenotype in the future to correspond to what's going to be optimal for the cell to grow. And therefore, there's going to be a possibility that having this slightly branched type of exploration of the future environment might be something closer to optimal than these other two. In bacteria, I realized I wasn't able to give that enough, do any justice to that. So I'm now going to conclude that I have no idea what I'm doing. But lineage distance seems to correlate with phenotype in the population level growth arrest transition. We need something like a diversity index to understand from the cell's perspective what's going on. And the thing that I skipped over was where I did a more rigorous analysis showing in certain restricted conditions that bet hedging may optimize memory, thus allowing a lower cost in the future environment. So I'd be happy to talk about that if you're interested. But then I'd just like to thank Arnab, has been working on these lineage simulations. I had collaborators and contributors to the first part of this project there, and that's the rest of my lab at this time, and I was funded by a Developmental Research Project Award.
All right, so we have time for a question or two while Ramon is setting up. So if you want to come up and start setting up, that'd be great. So any questions?
So Christian, do you have the toxin, antitoxin systems on plasmids or the chromosome?
So there are two types. There's a type where where one of them appears on a plasmid and the other on the chromosome, and then there's the type where they both appear on the chromosome. The ones I'm primarily concerned with, the toxin and antitoxin tend to be in the same operon inside in the chromosome of the cell.
And I mean, you engineer them so that you have control over the promoter and the promoter induction, in principle. So I mean, are they constitutive promoters?
Well, they're not constitutive promoters in reality, but I still have the microphone on. They're post-translationally regulated usually. So it's what happens when the stress occurs is that the antitoxin is degraded and it frees up the toxin that's already present in the cytoplasm.
Okay. Okay, what are we saying, Christian, again?